Welcome everyone. Thank you for joining us for our presentation titled Investigating Genetic Diversity in the VA's Million Veteran Programme, or MVP. It's my absolute pleasure to introduce you to Dr. Saiju Pyarajan, who is Director of the Center for Data and Computational Sciences at the Veteran Affairs Boston Healthcare System in the USA. For a complete biography of our speaker, please visit the tab at the top of your screen. If you have any questions during the presentation, we really encourage you to submit them in the Q&A box. Our speaker will address your questions following his presentation. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top of the presentation window, and please follow the process to obtain your credits. Please join me now in welcoming Dr. Pyarajan. I will now turn the presentation over to him.
Thank you for the introduction, and hello everyone. First, I would like to thank the organizers for giving me this opportunity to present today. Polygenic risk scores have recently shown a lot of promise as a way of predicting risk for complex traits based on weighted averages of the individual risk alleles. Clinically, PRS scores have begun to be used for risk stratified genetic screening, basically deciding on who needs to get genetic screening for a particular disease. It also is helping refining the individual risk assessment, which helps guide the kind of risk reduction interventions that can be taken. For example, in the clinic, it is helping to decide on the tailoring of the therapy, as well as changes in the lifestyle and environment, for example. It also is helping predict health outcomes, which is beginning to be used in complex diseases like breast cancer, prostate cancer, and diabetes. While polygenic risk scores is showing a lot of promise, there are a few challenges. As you know, polygenic risk scores are very dependent on the availability of very good quality GWAS summary statistics from a lot of samples across a lot of populations. We need good effect size and frequency calculations for the different predictions. One of the problems that we have been running into is the generalizability of these risk predictions across multiple populations. Alicia Martin and Mark Daly recently showed very nicely in a Nature Genetics publications, where they looked at the accuracy of prediction of PRS across European populations for 17 quantitative traits across 5 different populations in the UK Biobank. As you can see in the figure on the right, the prediction works really well for European populations. But for the rest of for the other ancestry populations, it is kind of still a work in progress, if you will. That brings us to the Million Veteran Program. The Million Veteran Programs was initiated in 2011 by the VA as a way to explore how genes, lifestyles, and exposure affect health and illness. We collect, as part of the Million Veteran Program, we collect biospecimens that are then linked to the EHR and self-reported and other health data. To give you a glance of what we have done so far, the MVP has collected over 828,000 samples. The collection sites are all across the United States. We have roughly about 61 roving collection sites that moves from station to station, depending on the interest shown by the veterans to the mailers that we sent about the MVP program. So to give you an overview of the MVP data universe, from the MVP participants, we collect self-reported surveys. We essentially have 2 types of surveys that we send out. One is a short 5-pager that asks questions about their health in general. And then we send out a much longer 15-page surveys that is more of a lifestyle survey. We collect 10 ml blood as a biosample from each of the participants when they sign their HIPAA and consent. And from these biosamples, we generate multiple molecular data that can then be integrated with clinical data that we get from the EMR, from the VA, the CDW, the corporate data warehouse that sort of abstracts all the clinical data from the EMR system. So this provides an opportunity to integrate longitudinal clinical data with their molecular data. We also get data from other sources, including Medicare, various disease registries, National Death Index, etc. So how do we do biobanking in MVP? The recruitment to MVP is basically an all-comers. The inclusion criteria is that you have to be a veteran and user of the VA healthcare system, and there is no exclusion criteria, so to speak of. We collect 10 ml of blood in an EDTA vacutainer at the time of recruitment. These blood samples are shipped overnight on ice packs to the central repository in Boston. Most samples arrive in 24 to 48 hours. Accessioning is done into the lab's LIMS system without any clinical identifiers. We save multiple aliquots of plasma, Buffy codes, and an aliquot of the Buffy code is then made into DNA, which is then sent out for molecular analysis. We take care not to have any sorting biases, and that's why not having any clinical identifiers in the LIMS system helps. Because this is not, we are not collecting, it's not a case control study, or we are not collecting any disease specific, so it's an all-comers, and for the labs, it is a sample, and that's it. So to start to look at the sort of the genomic analysis, we first developed an MVP genotyping array. We developed this with the help of Affymetrix or Thermo Fisher on the Axiom platform. We use the Axiom biobank as sort of the backbone onto which we added about 100K custom SNPs, mainly the SNPs that are clinically relevant. Over the next few slides, I'll just go through very briefly, the kind of design decisions that we took when we built the MVP custom array. As you can see, the backbone has the Axiom biobank content, which includes the pharmacogenetics, exome grid, loss of function, GWAS, coverage, SNPs, etc. On the custom content, we included a lot of clinically relevant SNPs, including sort of disease diagnostic or prognostic disease relevant SNPs, including RAs. We also included a lot of HLA and KIR region SNPs with the idea of being able to see if we can map these regions better. We also used increased sort of the, have tag SNPs that allows us to increase the imputation quality in different ethnicities. In terms of the clinical relevance of the MVP array, this is an example where we are looking at the top 10 ACMG genes and how the number of SNPs that have been used on the microarray to cover them. We pretty much have all the entire ACMG list is probed on this array. We also, as I said earlier, that we have been typing a lot of SNPs that are sort of important for disease groups. And here's a list of examples of the research areas and the diseases that have been probed by these markers. So this is an overview of the sample and data processing workflow in MVP. So we start with sample collection, we do the data extraction in our biorepository, and we send it out for genotyping. Since MVP is continuing to enroll participants, and the analysis is done over a longer period of time, we pay particular attention to how batching is done. So we make sure that the samples that are collected together are assayed together on the Axiom platform. With Thermo Fisher, we have developed a lot of advanced QC methods, including plate normalizations, sample quality control, sample and marker quality controls, and also methods to sort of minimize batch effects. We do multiple data quality control assessments, and then we also generate sort of secondary data in terms of relatedness, genetic ancestry, etc. And we validate the sample, the data before using, height as an example of a GWAS before making the data available for analysis. As I mentioned earlier, the MVP array has a lot of low frequency alleles because they are clinically significant markers. As you can see on the left, the missingness increases, the different buckets of missingness on the x-axis. You can see as the missingness increases as we have more rare alleles. On the right, you can look at the, what I'm showing you is sort of the minor allele discordance. As you can see, for the common alleles, 1% or above, we have the cumulative fraction of markers in that bucket, the allele discordance is very, very low. So even with for the allele frequency bins of less than 0.001, we have more than 80% concordance. Another quality metrics that we look at is duplicate sample concordance. As part of our analysis plan, we send about 5% of the samples back to the vendors without telling them that these are duplicate samples. So on the left, you can see the absolute concordance between those duplicate samples within the vendors and across vendors. It's pretty close to 100% and above 99%. And on the right, you can look at the minor allele discordance. Again, between vendors and within vendors, the minor allele discordance rate is very low. One of the things that we did is that we compared the allele frequency of MVP as a whole first to the 1,000 genomes. So in this, you can see the different minor allele frequency bins, and the MVP is a gray bar, and the 1,000 genome ancestry populations are the different colored bars. And as you can see, MVP has the MVP allele frequency is spread across the entire data set. What we have noticed is that if we split the MVP into the ancestry subsets, for example, the allele frequency of MVP seems to be different from the 1,000 genome, indicating that there is a lot of diversity in the MVP population. We also have done a lot of work on identifying related individuals to make sure that when the data is used for GWAS, we get good quality, GWAS analysis results. So we have used the kinship analysis to differentiate between, for example, parents, offsprings, full siblings, and first cousins. And we are also able to pick up duplicates or twins in that process. We have also looked at runs of homozygosity. As you know, the bottleneck populations should have longer runs of homozygosity, while the admixed population should have shorter runs. And we can see that the range that we have in MVP, indicating that we have populations across the spectrum in terms of admixture. Here's what I'm showing you is the projection PC, where we projected the MVP, which is in gray, onto the 1,000 genome population, with the PC1 and 2 on the left being shown on the left. As you can see that, yes, MVP does have the Caucasians and the YRI and the Asian cluster, but there is a pretty good spread of MVP samples between those clusters, indicating, again, the diversity that we have in MVP. And on the right is just another way of showing you the same thing, where gray is still the MVP. Another way of looking at genetic diversity in the MVP sample, about 70% of the MVP samples are clustered with the GBR population, for example. But you can see that there is a large number of population that cluster with YRI and other various sort of admixed population, so to speak. So this gives us an ability to have reasonably large numbers of each of those subsets of ancestries, which should really help us in doing GWASs within those subsets, rather than doing across all the genetic ancestry subsets. So in conclusion, MVP is very large. We have more than 800,000 already, and we are still continuing to recruit. And as I showed you, it is very diverse cohort and should enable genetic analysis across stratified populations, meaning that, even the minority populations, we should have enough in number to be able to do statistically relevant conclusions from GWAS analysis, for example. We have very rich and longitudinal clinical data that is associated with these samples, and which allows us to do very fine phenotyping in terms of disease and subsets of diseases. MVP data in general is of very high quality. And overall, what I want to, as a take home, what I want to say is that, MVP cohort will allow us to increase the scale of non-European populations on which genetic analysis has been done. This should really allow for building population subset specific PRS and even disease specific PRS, because we have enough number. The N should allow us to do meaningful statistical conclusions. And with that, I want to sort of acknowledge all the peoples involved in it, especially the MVP participants for their altruistic donation of samples and their data to the MVP program. Thank you, everyone.
Thank you very much, Dr. Pyarajan. Really great presentation from a very, very important study, the Million Veterans Program. We now intend to move into the live Q&A session. As a reminder, please submit your questions via the Q&A box. Just a note as we move into this session, of course, technology is wonderful when it works. Right now, we have a slight challenge that Dr. Pyarajan is in Boston and they've suffered power cuts. So he's running on battery power, and we have a plan B if we lose him on his current computer. But please do bear with us if we need to make that swap. Thank you very much. Dr. Pyarajan, are you with us ready for Q&A?
Yes, of course, yes. Again, thank you, everyone. I'm ready.
Fantastic. Okay, so we'll take our first question then that's been submitted. And again, I encourage the audience to please submit any further questions that you think of as we go. So our first question, Dr. Pyarajan, other studies like these kinds of prospective biobank studies have reported difficulty in recruiting participants from ethnic minority and vulnerable populations. What was your experience recruiting for MVP?
That's a great question. That's a great question. So when we started, we sort of wanted to recruit, as I said earlier in the talk, that it was all comers. Anybody who is a veteran and has used the VA system for their clinical care we welcome them all. Compared to the general population, the VA population is slightly skewed in terms of being more male, and it's also has a little bit more diversity across the ethnic population subsets. But as I said earlier, again, that the veterans are very altruistic, and anything that they can do to help their brethren, it's a good cause to put in, to throw in their hat. So we actually, the way that we recruited, initially when we started recruiting in 2011, we were using basically a mailer, a snail mail, US Postal Service mailers, and our recruitment rate is pretty high. So in general, we haven't targeted any subsets per se, but we have been able to, just by the type of population that the veterans is and their altruistic nature, we have been able to sort of recruit more of the minority populations.
Thank you. Thank you very much. So we can move to our second question, a little bit more around the technology. So it's been suggested that genotyping arrays do not perform well for rare allele genotyping, but your data suggest otherwise. Can you comment on this, please?
That is correct. Using, genotyping rare alleles is difficult, but we have taken a lot of steps to ensure that we get a very good quality genotyping, even for the rare alleles. For example, what we have done is that working with Thermo Fisher, we have arrived at a sort of an optimum size in terms of the batches that we do, that we process during our APT runs, for example. So we process about 50,000 samples in one go. So we have the computing power to be able to do that, and that allows us to pick, have more than one hits for some of the rare variants. We have done a lot of other things, including, once we run the APT, we look at the allele frequency, and then we compare the allele frequencies that we get to sort of other biobanks like the UK Biobank, the 1000 Genome, and the Nomad, to make sure that some of our allele frequencies are not way off. We have developed processes to look at the sort of the cluster plots to make sure that we do some rare hit adjustments when things don't look very good. We also have sort of included, a process of doing a Fisher's exact test that was developed in the UK Biobank to make sure that we don't have too many batch variations across the batches. As you can imagine, we are in the 800,000 rate, so even with 50 plates, we have a very large number of batches. We want to ensure that the data quality or the batch effect is minimal across all the batches. So with all those sort of methods that we have developed in these large data sets that we have been able to scale in this large data set, we are pretty confident that most of our genotypic calling is accurate and stable.
Mm-hmm. Great, thank you very much indeed. So a big piece of the design of the study seems to have been the selection of these 100,000 custom markers that you mentioned. So the next question is, how did you decide which specific SNPs of all the ones that were out there that you should include on that final list of 100,000 custom MVP SNPs?
Right, very, very good question indeed. So yes, we spent a lot of effort at the time of developing the chip to decide what we need to put in the custom, the custom part of the array. Once Thermo Fisher sort of was able to allow us to put in these custom SNPs. So at that time, one of the things that was not generally done with genotyping array was to sort of look at clinically relevant markers that may not are found to be clinically relevant in sort of different experiments that small experiments that people have done across the board, and some of them get into databases like the ClinVars or DBSNP and dbGaP, etc. So what we did is, including surveying these larger databases for clinically relevant markers, we sort of understood that our numbers are not going to be 1 million. We already had planned to go beyond 1 million if we were successful. So we were not worried about worried about including sort of very rare markers, which we may not get enough number, for example, in the whole data set. So our emphasis was to sort of add clinically relevant markers that can be used for either diagnosis or prognostic purposes. We also wanted to include these markers that are sort of have a different allele frequency in the different minority subsets. So we try to include them without sort of any sort of bottom cutoffs for the allele frequency, for example. And then we have sort of stressed on the diseases that are A, relevant to the veteran population, and B, some of these are more specific to the veteran population, or they are larger number in veteran population compared to the outside population. For example, tinnitus, which is a problem across the board in normal general population, but it is a little bit more enriched in the veteran population. So we included, for example, a lot of markers on tinnitus and sort of rare diseases, if you will. So that's what we did mainly for including, for developing those subset of markers that can be customized.
That's great. Thank you very much. We have time for maybe one or two more questions. So a great question coming up next. Will the genotype data be imputed? And if it will, perhaps you could also expand on the question by explaining how you factored that into the design of the array that you're using.
Absolutely, a great question. Yes, we have, in the last release of the data that we did to the users, we actually already imputed it to using the reference genome of the 1000 genome phase 3. So we always intended to impute it because the whole genome sequencing, though we intend to do whole genome sequencing, it is much more expensive, and this the array provides us a very cheap way of sort of screening across very large population sets with very large number of samples. So we have included a lot of markers, for example, for the minority populations, tag SNPs, for example, for the minority populations, as well as the AFI's biobank backbone that allowed for better imputation in those minority populations. Mm-hmm. And for the current release, we are planning to again, we will do the imputation, and this time we are planning to use the African genome reference panel, so that we are able to impute a little bit more better the African American population, for example, besides the Caucasians.
Excellent, thank you very much. So we should wrap up now. I think I need to check with you, how is the battery life on the computer, and do we think that the expert lounge is a viable option?
I am running pretty low. Unfortunately, we had a high winds here overnight, and we lost power. So no, we are not looking too good, but I'll try anyway. But we are not looking too good.
It is what it is. It is what it is. No problem at all. So I have to thank you again, Dr. Pyarajan. Thank you very much for your presentation. Thank you for taking the questions from the audience. Thank you to the audience for your questions as well. As a reminder, any questions that were submitted and not answered today will be followed up via email. This presentation will be available on demand through next year, actually. So please do share it with a colleague who might be interested in today's topic and the research that Dr. Pyarajan and his team are doing. I'll now close the presentation. With that, thank you and goodbye.
Thank you. Bye-bye.